Hello everyone, I welcome you all to today's video. Today we will discuss chemical bonding in biochemistry or chemical bonds that hold biomolecules together. I have already explained what is biochemistry and what biomolecules in my previous video. We are discussing this before understanding the types and structures of biomolecules because I want you all to understand the chemistry part of chemical bonding which is required in biology. Let's begin. Bond is a connection that holds things together. Chemical bonding is that connection that holds atoms together in a molecule. That is, the atoms are held by chemical bonds in a molecule. This happens because all the atoms they tend or have a tendency to attain the most stable form. For example, we all like to lie down in our bed and do nothing. That is our most stable form. So the atoms, they always have the tendency to acquire their duet or octet form. That is their most stable forms. A duet form is seen in case of hydrogen and helium, where they have 2 electrons in the outermost shell, and this is the most stable form of hydrogen and helium. Other atoms, they tend to have 8 electrons in their outermost shell, and this is the stable form of other atoms. So in by the help of chemical bonding, the atoms, they attain their duet or octet state. So when atoms, they attain stability, they form chemical bonds and convert themselves into molecules. In some cases, they donate their extra electrons, and in other cases, they share electrons. Over here, for example, this sodium is having 1 extra electron and chloride is having 1 deficiency. See, 5 plus 2, 7. And over here, if it will donate, this sodium will donate this extra electron, then it will attain its stability. So sodium is donating 1 electron and this is accepted by chloride. So sodium and chloride, they both are attaining stability. And during this process, bond is formed and they are converted into molecules. Over here also, oxygen is having a deficiency of 2 electrons and hydrogen is having a deficiency of 1 electron. So oxygen is bonding itself with 2 hydrogen atoms. Over here, this oxygen is sharing its electron with hydrogen, as a result of which hydrogen is having 2 electrons, that is it has attained its duet. And over here also, this hydrogen atom has attained its duet. And this oxygen, because of this sharing, has attained its octet, as it had 4 plus 2, 6. So it needed 2 electrons. Hydrogen had 1, so it needed 1 electron. Over here, it got 1 from this oxygen, and over here, this hydrogen atom also got 1 electron from this oxygen atom. As a result of which, all the 3 atoms have attained stability. They are converted into molecules because of chemical bonding. Let's see the types of bonds which are required to understand the concepts in biology. There are basically 2 types of bonds: intramolecular bonds and intermolecular bonds. The word intramolecular means inside molecule. Let's suppose water we are taking as an example. So it is a molecule formed due to bonding in between 1 oxygen atom and 2 hydrogen atoms. So when we are talking about the interaction between this oxygen and these 2 hydrogen atoms, then those interactions are called as intramolecular interactions. There are 2 types of intramolecular bonds, that is covalent bond and ionic bond. Similarly, the word intermolecular means in between 2 molecules. So let's suppose we are taking 2 water molecules. So what will be the interaction between these 2 water molecules when they will be present together in a container? Let's suppose we are having a glass of water. So what will be the interaction in between the water molecules present inside that glass of water? Those interactions are called as intermolecular interactions. And we will deal with basically 3 types of intermolecular interactions, that is hydrogen bond, dipole-dipole interaction, London dispersion forces. We will also discuss about few special types of bonds like Glycosidic bond, Peptide Bond, Phosphodiester Bond, disulfide bond and Hydrophobic interactions. When we will come to know the meaning of all the words which are written on the board, then everything would be clear in relation to biochemistry. Now we will see the types of intramolecular bonds, that is ionic bond and covalent bond. what type of bond will be formed in between atoms in a molecule will be decided by electronegativity. Electronegativity is the power of an atom to attract the bonded pair of electron towards itself. To understand this, let's take an example. Let's suppose A and B are the atoms which will form bonds to form a molecule. Let's suppose A and B have the same value of electronegativity. A and B are having the same control over this bonded pair of electron. That is, A and B will not exert different powers on this bonded pair of electron. So this shared electron will be present in the middle of A and B, as a result of which the bond which will be formed will be called as pure covalent bond. So covalent bond is formed due to sharing in between two atoms, sharing of electrons in between two atoms, and that too, those two atoms will have same values of electronegativity. Let's suppose A is having electronegativity of 3, that is 3 is the value of electronegativity of A, and B is having electronegativity power of 2.7. So they are almost same, or the difference in between them is less than 0.4. If the difference in electronegativity of two atoms involved in a molecule is less than 0.4, in that case the shared pair of electron will be present in the middle, and both the atoms will exert the same amount of force, as a result of which it will not be attracted towards any one atom in a molecule, and the bond which will be formed will be called as pure covalent bond. This type of bond can also be called as non-polar covalent bond, as covalent bonds are of two types: polar covalent bond and non-polar covalent bond. A polar molecule is a molecule in which the atoms involved have some amount of charge, as this electron is shared equally in between A and B, so A and B develop no charge. Let's suppose this electron pair is attracted more towards B, as a result of which A will develop slight positive charge and B will develop slight negative charge. But over here they both are having the same value, almost same value of electronegativity, so they both are neutral, that is, they both have not developed any amount of charge. So this molecule will be non-polar, as there are no poles developed. Poles mean two ends of a molecule, and those two ends will have different charge. There will be a charge separation at in those two atoms in the molecule. So this bond will be also called as non-polar covalent bond. The examples are hydrogen molecule, chlorine molecule, chloride molecule, or chlorine molecule you can say. Hydrogen molecule is formed due to non-polar covalent bond in between two hydrogen atoms. These two atoms have same electronegativity power, so they will share this electron pair equally. Similarly, two chlorine molecules also will share their electron pairs equally. In second case, let's suppose B is having a higher value of electronegativity than A. Let's suppose B is having a value of 5 and A is having a value of 3. In that case, B is having more control over the bonded pair of electron, that is, it is having higher electronegativity, so it will attract that pair of electron towards itself. It will be shared in between A and B, but in that case B will have more power, more control over that pair of electron. Over here I have written the electronegativity power of B is greater than A. Over here it was A is equal to B. In that case, the bond which will be formed in between A and B will be called as polar covalent bond. Polar covalent bond means due to attraction of this electron more towards B, B will develop a slight negative charge and A will develop a slight positive charge. Because A is in loss, it is losing that electron pair, and B is gaining that electron pair, it is having more power, so it will develop a slight negative charge. And whenever the charge is not of higher magnitude, there we are writing a delta, we are using the word delta. So B is having delta negative charge and A is having delta positive charge. So this molecule will be a polar molecule. There are two poles, a slightly positive pole and a slightly negative pole. and this electron is shared. So this will be an example of polar covalent bond. The examples will be HCl or H2O. In case of HCl, the electronegativity power of chlorine is greater than H, so it will attract the electron pair towards itself. Similarly, in case of H2O, O will attract the electron pair towards itself more than H. So O will develop slight negative charge, H will develop slight positive charge. Similarly, Cl will develop delta negative and H will develop delta positive. Over here, dipoles are also formed. Dipole means a molecule where two poles are developed. That will be a dipole. So this is a dipole, as this is a positive end and this is a negative end. So polar covalent bond also leads to the development of dipoles. In third case, A and B, they have a large difference in electronegativity. Let's suppose A is having the value 3 and B is having the value 7. In that case, B will have more control over that pair of electron and it will attract that electron pair towards itself completely. And A will have zero control, B will have full control. Over here, A had somewhat little bit control was there, but B had higher control. But over here, A is having zero and B is having full control. As a result of which, the bond which will be formed between A and B will be called as ionic bond. Now we will see the types of intermolecular bonds. The first one and the most important one is hydrogen bond. Over here I have drawn one hydrogen bond. This is a water molecule, this is another water molecule, and these two water molecules have hydrogen bond formed in between them. So this is a type of interaction in between two molecules, that is intermolecular interaction. The hydrogen bonds are represented in the form of lines. Hydrogen bond is a bond formed in between two molecules, and those molecules will have H, and either that hydrogen will be bonded with fluorine or oxygen or nitrogen. So hydrogen bond will be formed in between two molecules in which these two molecules will have H. Hydrogen will be present compulsorily in these both molecules, and along with H either O will be present, nitrogen will be present or fluorine will be present. And when such molecules will come together or come close to each other, then hydrogen bond will be formed. For example, ammonia, the bond formed between two ammonia molecules will be a hydrogen bond, because over here hydrogen is bonded with nitrogen. Similarly, the bond formed between two hydrogen fluoride molecules, or the bond formed between two water molecules, or the bond formed between ammonia and HF, or HF and water. So the bonds which will be formed between such molecules will be called as hydrogen bond. See over here, this oxygen atom is bonded with two hydrogen atoms. Over here I have drawn the bond as well as the electrons to make it clear. Don't get confused. Either you can draw bond or you can represent electrons. So this oxygen is highly electronegative as compared to hydrogen. So the bonded pair of electron, this electron is shared in between hydrogen and oxygen. So this oxygen will attract or will have more control over this bonded pair of electron. So oxygen will develop a slight negative charge, delta negative, and hydrogen will develop a slight positive charge, that is delta positive. And the bond which will be formed due to this sharing, this unequal sharing, will be called as polar covalent bond. So in water molecule, inside water molecule, in between hydrogen and oxygen atoms, the bond which will be formed is polar covalent bond. Sharing is there, but it is unequal and poles are developed. When such sharing occurs, this molecule will be called as a dipole. This molecule will interact with another such molecule, as a result of which the bond formed will be called as hydrogen bond. This hydrogen bond is formed because of this lone pair of electrons present on oxygen atom. There are two lone pair of electrons, and the hydrogen atom of another water molecule. This hydrogen atom is having very little control over this shared pair of electron. That is, it is having very little role in this bond which is formed due to that shared pair of electron. So the bond can be easily formed in between these lone pair of electrons, or this interaction can easily occur in between the lone pair of electrons of oxygen and hydrogen of another molecule. such bonds in between molecules having hydrogen and another more electronegative atom than hydrogen, that is fluorine, oxygen and nitrogen, is called as hydrogen bond. Hydrogen bond is the strongest intermolecular bond, but it is a very weak bond as compared to covalent and ionic, that is, it is very easy to break this type of bond. But when we will compare different types of intermolecular bonds, that is dipole-dipole interaction, London dispersion forces and hydrogen bond, hydrophobic interaction, when we will compare all the different types of intermolecular bonds, in that case this is the strongest. But when we will compare it with intramolecular, that is when we will compare this bond with this one, it will be much more difficult to break this bond as compared to this bond. So now we will see the other two types of intermolecular forces, that is dipole-dipole interaction or dipole-dipole forces and London dispersion forces. There are other types of intermolecular interactions also required in chemistry, but for biology purpose, if you will understand these two forces, then it would be sufficient. These two are the types of van der Waals interactions or van der Waals forces. That is, these two are subtypes of van der Waals interaction. This is a very weak type of interaction, and this is an interaction between electron clouds of two molecules. That is, suppose two molecules, they come close and their electron clouds somehow interact. So that type of interaction is van der Waals interaction, and there are many different types of such interactions. The first type can be dipole-dipole interaction. As you all already know, dipole is a molecule where there is a charge separation, that is there are two different poles, a slight negative and a slight positive. So let's suppose an example, a molecule HCl. Let's suppose the electron cloud of HCl interacts with another HCl molecule. So the electron clouds of these two molecules, if they interact, that type of interaction will be called as dipole-dipole interaction. Over here, hydrogen is the slightly positive pole and Cl is the slightly negative pole. So this is a dipole. This dipole can interact with another dipole, that is, this slight negative end can form a bond with the slight positive end of another such molecule or any other dipole molecule. So a dipole molecule, if when interacts with another dipole molecule, that is the interaction takes place in between two different poles of two polar molecules, that case is called as dipole-dipole interaction. We have already seen hydrogen bond. In hydrogen bond, what happens? This oxygen interacts with hydrogen, and water molecule is also a dipole. Another water molecule is also a dipole. So hydrogen bond is also a type of dipole-dipole interaction. The difference between a pure dipole-dipole and hydrogen bond is that hydrogen bond is formed only between two molecules when the molecules have H and F or oxygen or nitrogen. So the molecules which are involved in the formation of hydrogen bond should have hydrogen along with any one of these atoms. So hydrogen bond is a special type of dipole-dipole interaction. But the interaction between any two dipoles, any two molecules which are dipole, is called as dipole-dipole interaction. The other type of van der Waals interaction is London dispersion forces. This is also called as instantaneous dipole-induced dipole. The other type of van der Waals interaction is London dispersion forces or instantaneous dipole-induced dipole. Van der Waals interaction is a type of intermolecular interaction, and it is of many types, but we are discussing only two types over here. In this case, let's suppose two hydrogen molecules have come together and their electron clouds are interacting. That is, let's suppose in case of hydrogen molecule, the electrons which are rotating continuously around the nucleus in their orbitals, let's suppose for a moment or for a very, very short period of time, all the electrons have gathered towards one end of that molecule. So the end which is having the electrons gathered for a very short period of time will develop a slight negative charge. The other end of the molecule will develop a slight positive charge, though this condition is only for a very short period of time. But for that short period of time, this non-polar molecule has developed a slight polarity, but that polarity is for instantaneous moment. It is for a very, very short period of time. But this non-polar has converted itself into a polar molecule. So this non-polar molecule has converted itself into a dipole, and if such dipole comes in contact with another hydrogen molecule which is normal, which is not undergoing such scenario, but due to this dipole, instantaneous dipole, this hydrogen will be converted into an instantaneous dipole. That is, the positive end will interact with this positive end will force the electrons to gather at another end of this second molecule. So this interaction will be called as instantaneous dipole induced dipole. This instantaneous dipole will induce dipolarity in the other molecule. Such type of interaction will be called as London dispersion forces. Now we will see some special types of bonds, the meaning of which is required to understand biochemistry. The first one is a peptide bond. Peptide bond is a type of covalent bond formed in between amino acids. As you all know, amino acids, they form peptide bond to form a protein. That is, you can compare amino acids with the beads, and the beads, they come together to form a necklace. So the necklace is protein and amino acids are the molecules which are the beads, and amino acid is a type of biomolecule which I have already told. So the bond in between two amino acids is called as a peptide bond, and it is a type of covalent bond. How peptide bond is formed, we will discuss in our upcoming videos. I am just telling you the names so that you can just remember those things when we will discuss it actually. A disulfide bond is a type of covalent bond, same. It is a type of covalent bond formed in between two amino acids, but those two amino acids are a type of amino acid which is called as cysteine. As you all know, there are 20 important types of amino acids in this world. Every living organism is having only 20 amino acids. Those 20 amino acids, they combine together in different combinations to form different types of proteins. A type of amino acid is called as cysteine. This cysteine is having a sulfur atom. So the peptide bond which is formed in between two cysteine amino acids is called as a disulfide bond, which is also a type of covalent bond. The next type is hydrophobic interaction. Hydrophobic interaction is an interaction between water molecule and non-polar molecules. This type of interaction is seen in case of proteins. Peptide bond is also seen in case of proteins. Disulfide is also seen in case of proteins. Hydrophobic interaction is mainly seen in case of proteins. It is an interaction, let's suppose there is a protein and there is a non-polar amino acid. Now you all know the meaning of non-polar, that is a molecule where there is no charge separation. So that non-polar amino acid will interact with water molecule which is present in our body. That interaction, that because of this water, the non-polar amino acid will try to move away from it. So the protein will be folded in such a way that the amino acids which are polar in nature will be present outside, and the non-polar amino acids will be present towards the interior of the protein because of water molecules which are present surrounding that protein molecule. Such type of interaction is called as hydrophobic interaction. It can be also compared with a drop of ghee or oil in a glass of water. That type of interaction is also hydrophobic interaction. The next type of bond is a phosphodiester bond. It is also a type of covalent bond. This phosphodiester bond is seen in case of DNA molecule. What is it and how is it formed? How it is a type of covalent bond will be discussed when we will discuss the structure of DNA. The last type of bond which is required in biology is a glycosidic interaction or glycosidic bond. This is also a type of covalent bond. A glycosidic bond is formed in between two carbohydrates, or a carbohydrate and a non-carbohydrate molecule. That is, let's suppose a glucose molecule, if is bonded with another glucose molecule, that type of bond is called as glycosidic bond. Similarly, let's suppose ribose sugar, that is pentose in case of DNA, is bonded with a purine. Purine is a nitrogenous base. That type of interaction is also called as glycosidic bond. We will discuss these things in clarity, that is both in between two glucose molecules or in between a ribose and nitrogenous base. How is it formed and how is it a type of covalent bond? We will see these things when we will discuss carbohydrates in detail. With this, we come to the end of this video. In our next lecture, we will talk about carbohydrates, then we will see amino acids, proteins, DNA. We will discuss all the topics of biochemistry one by one. I have discussed these things with respect to biology. I am not a chemist, I am a biologist. So I have given a touch of biology and I have tried to justify this topic as much as possible. Thank you for watching.